The following audio recording has been designed for learning purposes only. Do not share, distribute, or provide any type of copies, or this audio lesson will not be made available to you. Good luck with today's lesson.
All right, so the first test is always the worst. What'd you say? The first test is always the worst. You get the learning experience. So I'm going to have an extra credit assignment for you coming up. I'd like to get through these chapters first, though, so that we can move forward then. Is it? It's not that it's harder, it's just the fact that there's no labels. There's no book with you. You just got to know the stuff.
Yeah.
Okay? And you guys are actually lucky that it's multiple choice. When I took anatomy, it was a blank sheet, and they said, write it. And write it correctly, or we're going to take half a point off. So you do have a little bit easier than the old days. All right. But the first tests are always the most difficult because you don't know what to expect.
Yeah.
Okay? So, now you do. Okay. I wasn't kidding on the first day where I said this is a pretty tough class. You're going to have to put the time in outside of class. You really do. You've got to study this every day. You've got to go through some material every single day. Questions about, not your specific grades or anything. We can't talk about that. We need to do that privately. But any questions about anything else? It was there. There it goes. It's a bright day, and there's only so much light we can get out here. I could try shutting it back. I know. See if that helps a little bit. Okay, so we're going to kind of go through this chapter a little bit quickly because you probably had some of the material already from the lab manual and doing it in lab. So chapter 6 has to do with the skeletal system. Chapter 7. I'm sorry, I'm so used to the old book. I apologize. I'll get it out of my head one of these days. You know, after you do it for so long, it's just kind of ingrained in your brain. Yeah, I apologize. All right, so your bones and teeth are some of the most durable remains of a once living body. Ooh, how weird is that? Right? They will survive long after you're dead. Is that amazing? Yeah, they are very durable. But, you know, we don't want to talk about a dead skeleton. We want to talk about the living skeleton. Okay, and there are, like any other organ, several tissues that make up bone, right? We could consider bone, every single one of them, an organ. So we've got lots of cells, we've got nerves, blood vessels. If you've ever broken a leg, you know how much it hurts, right? The bone is damaged. But your skeleton continually remodels itself, and it also can interact with other organ systems like your muscles. So when you look at bone on a slide, right, when we do some histology of bone, and just even studying bone itself is osteology. And there are lots of doctors who do this. A lot of, anybody know what kind of doctors you would see if you had orthopedic doctors, right? They study bone. They know all the ins and outs of bone. All right, so again, every bone could be considered an organ because it's not made up of just bone. It's made up of some cartilage, some dense connective tissue, some adipose, some nerve tissues, lots of blood vessels. Yeah. So they're all working together, okay? Cartilage Cartilage actually is the forerunner of most bones. Yep, because if you were born with bones this big, your mom would never have a sibling for you. Right? Yeah. So as the fetus is developing, it needs sort of a model. How am I going to make a bone? How do you put the bone together? So we actually use cartilage as a model, and then the bone replaces the cartilage. Pretty cool. And cartilage is a little more flexible. Dense connective tissue, yep, we're going to have coverings around the bones. We are going to have ligaments that hold bones together. We're going to have tendons that attach the muscles to the bones so that we can move the bones. So all of these things come into play. So you already know about the appendicular and the axial skeletons, right? Any questions about that? Okay, then I'm going to move on. So what are the functions of the skeleton? Probably something you didn't learn in lab. Number one, support. Yeah, it holds up the whole body, right? It supports muscles. It's got a maxilla and a mandible with teeth in there. Your teeth are actually part of your digestive system. Yeah, pretty cool. We'll learn about those. Protection, yes. Your brain is a little thicker than pudding. Yeah, protection. But we also have protection around our spinal cord where we have lots of nerves. We have protection, ribs that go around our heart and our lungs to help protect those organs. So protection. Movement is pretty obvious, right? Mineral storage, yeah. We can store lots of calcium and phosphate ions there. What do we need phosphate for? Energy molecules, ATP. What do we need calcium for? Well, we need it to make bones hard, but we also need it for a lot of other things in the body, like muscle movement and some other nerve transmissions and all kinds of things. So we need a lot of calcium too. Within the bones, right, is the formation of blood cells, red blood cells. Yeah, we call that hemopoiesis, making red blood cells. When we talk about blood, we're going to find out they only last about 120 days. And then they have to be recycled, and we make new ones all the time. So right now, you're probably making, I don't know, 1 trillion new red blood cells right now. Okay? But we also can make some white blood cells in there, too. And bone marrow, right? As we get to be this age, some of our bone marrow becomes yellow marrow, which means it's going to store triglycerides or fats. So it's a place where we can store some fat. So we don't starve to death. That's probably why. How long can you last without eating? Yeah, I can't hear.
1 week.
1 week? Yeah, you might think that. Yeah. Anybody else?
3 weeks.
3 weeks? Yeah. Yeah, you could probably go, I don't know, 3, 4 months, depending on how much your body has stored stuff. Yeah. I mean, not that you want to, but you could. You could only go a day. I know. Me too. I can even go half a day. Are you kidding? Yeah. Bone. Bone is also referred to as osseous tissue. So we have this prefix, OSS, that usually goes along with bone. Okay? So OS, we're usually talking something about bone. Connective tissue. Remember we talked about tissues, but bone is a connective tissue, and it's got a hardened matrix. Remember tissues. Connective tissues secrete that matrix around them. Okay? And in this case, when the bone cells secrete the matrix, they become trapped in it. And then we have some mineralization or calcification that occurs where this calcium phosphate comes over and it makes the bone really hard. The bone cells secrete the fibers, a lot of collagen fibers. Have you ever seen them putting down the cement roadways? When you're driving by, you see they put all these little rods in there. Does everybody kind of know what I'm talking about?
Mm-hmm.
Yeah. That's kind of like the collagen fibers that the bone cells put down in bone. And then the big cement truck comes and it dumps the cement in there. That's like this calcium phosphate, along with a bunch of other minerals, that harden them, right? That rebarb, those little poles, help to make the cement stronger. Same thing here. The collagen fibers get laid down. We put a little cement, calcium phosphate over top of them, and that makes the bone nice and strong. Okay. Sound good? Questions or anything? All right, you got to interrupt me. Tell me to stop. Explain things again, okay? I want you to feel comfortable with going, yo, stop. All right? Whoa, whoa, wait. What are you saying again? Okay, please don't let me go on if you don't understand something. Okay? All right, so we classify bones by their shapes. Can you name a couple of flat bones for me? Sternum. The skull. All the bones of the skull are flat, yeah. Yeah. Long bones. Humerus, femur. Oh, even your little phalanges, right? Because they're longer than they are wide. That's what we call a long bone. Yeah. Short bones. Can you think of any short bones? No. The talus. All of the little. Some lovers try positions that they can't handle. Right? All the bones of your wrists. Yeah. And your regular bones. These are the ones that don't fit into any other category, like vertebrae. Those are really weirdly shaped. Okay. So here's just a little picture. Okay. Yeah. Okay. So we can classify bones that way. And, you know, the reason we classify them by their shapes is because they're made in different ways. Okay. All right. So we have two types of bone, compact and spongy. Okay. Compact bone, yeah, it's really dense, right? There's not a lot of space in between. Spongy bone is also called cancellulous bone. It's a little more loosely organized, and we call it spongy because it looks like a sponge that you have on your kitchen sink, right? Because you've got all these little spaces in between. Okay? So this irregular pattern, not patter, sorry, I still can't type, pattern, okay, of little thin columns that are called trabeculae. That actually means little beam in Latin. Trabeculae means little beam. So it's like all these little beams, I can't say it now, beams connected together. Okay. We find it in it's found in the center of the ends and center of the shafts of long bones and in the middle of nearly every other bone. Wherever we have spongy bone, though, it's going to be covered by compact bone. Compact bone is a lot more durable, it's tougher, it withstands some stresses a lot better than the spongy bone. But the spongy bone also can withstand some stresses that are put on the bone because it's got that irregular pattern. So it just depends on the type of stress that we're putting on it, right? Bending kinds of stresses, spongy bone. Stress like being hit on the side, standing up on their bones all day, yeah. Compact bone is a little better for that kind of stress. So the reason we've got these different kinds of bones is to withstand the stress that we put on the body. Okay. Yeah, I mean, there's some cool things like 3/4 of the skeleton is compact and only 1/4 of it is spongy bone. That's kind of cool. So obviously we've got a lot more compact bone, more durable bone than we do spongy bone. All right, so you've seen this before. Any questions about that? So even in the long bones, there's a little spongy bone covered by compact bone. Wherever there's spongy bone, it will be covered by compact bone. Right? So, you've gone through the structure of a long bone. Anybody have a question? Okay. I'm going to go right to this picture. So, this is considered the diaphyses, the long part, or what we call the shaft of a long bone. On both sides, there are epiphyses. Epi is a term that means above, on the ends, right? Mm-hmm. So epi, we're going to see that term a lot, right? Mm-hmm. Epi, epineurium, epimysium, epi on the outside, right? So we've got a proximal epiphyses, the part that's closest to the body, and then we have a distal epiphyses, the part that's on the other end. Yeah, I've never seen it used very much. Maybe an orthopedic surgeon would, but we've got that little transition between that skinny little diaphyses going into a big epiphyses. So they call that little area in between as it kind of goes from one to the other, a metaphyses. Okay. I've not seen that term used very much. The epiphyses are where we're going to make joints, where two bones are going to come together. And we're going to be able to do elbow stuff, knee stuff, finger stuff. Yeah, so we're going to have a little articular cartilage on both sides. And again, that's going to make it a lot easier for movement, and we're not going to destroy the bone. Right? Yeah. The cartilage is going to help to protect the ends of the epiphyses, where they're going to make joints with other bones. Okay. Spongy bone, that's where we have red bone marrow. That's where we're making the red blood cells. But even in the shaft, we're going to have a little bit of spongy bone on the inside, and then all of it's going to be covered by the compact bone. Within the medullary cavity, there's going to be a lot of blood vessels. Yeah. The bone cells are alive. They're not dead. They're trapped in their matrix, but they're still alive. So, yeah. And we're also going to have some, nerves and things that are going to travel through the bone cells. Yeah. So all of that's in here. We're going to have blood vessels up here and nerves up here and the spongy bone too. On the inside of the medullary cavity, there is a membrane, epithelial tissue, the endosteum. Endo means in, endosteum. On the inside. On the outside, we're going to have a periosteum. Yeah. There you go. So, what do we have making this up? Bone cells, nerves, blood vessels, connective tissues. That's why it's an organ. Each and every bone could be considered an organ. It's got several different types of tissues working together for a common purpose. Any questions about the, what would we call it? Structure, I guess, of the long bones. Now, and I know it seems funny, but that's the same structure that's in these little phalanges. Because it's a long bone. It's a small long bone, but it's longer than it is wide. Hence, that's why it's called a long bone. We usually show you a humerus or a femur because those are the big long bones that we think. I love the femur. It looks like a club, doesn't it? Yeah. It's really long. It's probably the longest bone in the body. Why would you think the femur is so long? It's holding all this up, yeah. It's got to be nice and strong. It's holding up all the stuff on top of it and all the organs in here, right? Not a lot of organs down in your feet. So it's holding all that up. It's got to be a really big, strong bone. Okay? So we're good with a long bone? Okay, anybody questions? All right. Flat bones. Flat bones are kind of like a sandwich. Yeah. There's compact bone on both sides with the spongy bone in the middle. And good examples of these, the flat bones, are your skull bones. And what do we have here? What's this bone? Frontal. You just learned lobes of the brain. How cool is that? Yeah. At least in the head area, we kind of keep the terms the same, so it makes sense, right? You had some muscles here too, right? Temporalis muscle over the temporal bone. You don't have parietal muscles, but you have that frontal muscle, the occipital muscle, what else? That's it on the top of your head. So this part up here, it's just bone and some ligament. That's why it breaks it really easy, doesn't it? Okay, you guys on Mondays are bad. All right. There you go. A lot of times they refer to spongy bone as diplo. I don't know why. It's probably a Latin word, and the first person who ever saw it said, oh, that looks like, I don't know, sponge cake? I don't know what. But they came up with this word. I just never took the time to look it up to see what it actually means. But it is that spongy layer that's kind of sandwiched within the two compact bone layers. Okay. So this is, as you become an adult, the place where all of our red bone marrow is going to reside. And that's where we're going to be able to make red blood cells, white blood cells, and whatever else our blood makes. And we'll talk about that later on. Okay. We do have an endosteum that kind of winds its way through all of the little spongy layers, right? It's endosteum. Okay. And, bone cells can be replaced. So we think that endosteum actually contains some bone stem cells. Okay. And in fact, we can repair a fracture. So we're going to need some stem cells that can become the bone cells and fix whatever gets broken. We think the periosteum has a few too. So inside and outside of the bone, we've got some cells waiting to be activated to become bone cells. All right, so you've seen all this in lab. Any questions? Yeah, these little things are called trabeculae. They look like little beams. You know how when they're building a house and you see all the framing that's going on? Yeah, you could kind of think of that. Looks like that framing. Yeah, like little beams, all pieces of wood everywhere. Only here we're talking about bone. What are the bone cells? This is probably something you did not get in lab. Okay. Well, we have 4 different types of bone cells. Osteo, O-S-S-O-S-T. Yeah, they all mean bone. So, like every other tissue we have to have some stem cells. So we call them osteogenic cells. So these are the stem cells. Now, these cells, when they are given the proper signal, become what we call an osteoblast. And then they're going to mature into osteocytes. So I think of osteoblasts as baby cells and osteocytes as the adult cell. Okay. It is the same cell. It's just at different stages of making bone. So an osteoblast is the baby cell making collagen fibers and excreting them, making the part of the matrix. And then once the matrix is made, and it gets hard, they are trapped in the matrix. And so they are going to then mature into an adult bone cell called an osteocyte. So the osteogenic stem cell divides, mitosis. The cell, one cell is going to stay a stem cell. The other cell becomes an osteoblast. It starts to function, secreting all the collagen fibers outside of it. Then we mineralize it. And now that osteoblast is trapped in that hard matrix. So it matures into an osteocyte. And it can still secrete things and help to repair things even if the bone isn't damaged, but it helps to maintain the bone. The fourth type of cell also comes from the osteogenic cell. It's called an osteoclast. You know, if you broke a bone, you're going to have some jagged pieces and some destroyed bone. How do we get rid of that? Osteoclast. They come over and they literally dissolve away broken pieces of bone. Yeah? And they make a nice smooth surface so the osteogenic cells can divide and the osteoblasts will come over and they'll start making new bone to replace where the break was. So they are going to help to repair bone or help us to remodel bone. Yeah, probably about 10% of your bones get remodeled throughout the year. Maybe even more, depends. So here we are. We start with the osteogenic cell. It undergoes mitosis, divides in two. One stays a stem cell, the other one goes on to become the osteoblast. The osteoblast has lots of functions, right? It's going to secrete all of that matrix outside, in particular a lot of those collagen fibers. So we're putting down those metal posts in the road. And then mineralization is going to occur and make it really hard. And then it becomes an osteocyte. When it becomes an osteocyte, it starts to create these long cell extensions because they're trapped. How are they going to get nutrients and oxygen and get rid of waste products? They're going to use these cell extensions to meet up with another osteocyte so they can pass things back and forth. This is an osteoclast. Again, when a signal is sent, several of the stem cells are going to actually fuse together and create this funky looking cell that looks like it has a comb on it. They call that the ruffle border. Once again, it's going to be able to secrete lots of things that will help to break down the bone, dissolve, right, the calcification or mineralization, as well as the old bone cells. And it starts to reabsorb some of the good things like the calcium and the phosphate so that they can return it to circulation so it can be used to make new bone. So if this was where the bone was broken, the osteoclast comes in, secretes some sort of acidic chemicals that help to break down all the bone and debris that might be where a break is. And then an osteoblast will come over and start to create new bone. And then once it's created the new bone, it's going to be trapped and become an osteocyte. This happens, oh, it's probably happening right now. Oh, it's freaky. Oh, my bones are breaking. So, the osteoblast, we call it the bone-forming cell because it secretes all of the matrix, all of the collagen and all the other materials that are required for the bone to become hard, right? And that's before calcification occurs. So anything that's required, these osteoblasts are going to secrete it, right? So again, they usually line up as a single layer of cells under the endosteum and periosteum. So what they're secreting, of course, we have to give it a name. They call it osteoid. So everything they secrete, the soft organic materials, the collagen, everything that's going to become hardened by the mineral deposition or mineralization, calcification, right, is going to be secreted by the osteoblast. The osteocytes are now going to become trapped in that, and especially once we make it really hard with the mineralization. So as they are trapped, they start to send out these little cytoplasmic processes. And they reach into these little canals, which again in Latin are canaliculi. And then two osteocytes are going to connect in one of those little canals or canaliculi, and they're going to be able to pass things back and forth. Because the bone cells are alive. They're not dead. They still need oxygen. They still need nutrients and vitamins, and they still need to get rid of waste products. So they're alive. And the only way they can do that, if they're trapped, is by passing things from one cell to the next cell to the next cell, until we can get it into a blood vessel. Or get something out of the blood vessels and pass it back the other way. There you go. This is an actual photograph. Here it is. That's a live osteocyte in its little cavity here. That little cavity is called a lacuna. It's like a little cave. It's the area around it. And then these are those cytoplasmic processes that are reaching out. Right? It's kind of hard to see the canaliculi here, but you can see where it's going through the bone here. Right? So there's a little canal in here. And those little canals are going to connect the osteocytes to each other. And they can pass things back and forth. Questions about the bone cells? Pretty good? Everybody make sense? Okay. There you go. Spongy bone. Compact bone. Spaces in between all of the bones, right? And in between the trabeculae. And that spaces are filled up with bone marrow. Does anybody know when they're doing a bone marrow transplant, do you know what bone they take the bone marrow out of? Hip bone. Right? That big flat ilium, yeah, it's filled with bone marrow. So they take this big ass needle, this big needle. Right? They do numb you up a little. But they take this big needle like this, and then they, yeah. Ooh. Gives me chills just thinking about it.
It's actually like a corkscrew when they do it.
Yeah, because they got to go through the
They have to
bone.
pull out like a cord.
Mm-hmm. Because the bone, right? They got to go through the compact bone to get to the spongy bone inside where the bone marrow is.
Yeah, that's your heart.
Woo! Yes. You can probably YouTube that if you're really interested. But we can see that the compact bone has a lot of different structure to it. Yeah, it's very dense. And there are the little osteocytes, and they're trapped in this hard matrix. So we're going to talk a little bit about how compact bone looks. Okay. So again, just I put some stuff in about the osteoclast too. We mentioned this. It's going to dissolve bone. Okay. Yeah. And this ruffled border, it looks like a comb to me. Yeah, comb in your hair. Yeah. And this is where we're going to secrete things, and they're going to be able to reabsorb a lot of the material so we can use them again, right? There's some collagen there, so if we break the collagen down, we've got amino acids because it's a protein, and that way we can reuse the amino acids to make more collagen. Mm-hmm. So, our bodies are really good at recycling things because, we go through a lot of hard work. We use a lot of energy to make things and digest things, and if we can recycle it some way and reuse it, why not? Yeah. Saves us energy, saves us time, keeps us from getting too hungry. So we mentioned lysosomes are like little acid vats. Yeah, lysosomes can dissolve the bone. So it's like acid. All right. So the matrix, yeah. By dry weight, it's about 1/3 organic and 2/3 inorganic. The organic matter is osteoid. There's some other things other than collagen, but we're not going to worry about all those. Okay, just mainly collagen, like they call it rebarb that they put down in the cement. Okay, so we're putting down all these collagen fibers over which we can put some inorganic material. That's called hydroxyapatite. And that crystallized calcium phosphate salt. Part of it is calcium carbonate, and there's 5% of other minerals like magnesium, and chloride's in there too. You don't need to know the breakdown, okay? But just recognize that we've got that osteoid, which is mainly going to be collagen fibers, and then we have hydroxyapatite that covers it like the cement. That's the best analogy I can kind of come up with for how we're putting down hard bone like that. All right. Now, what's really interesting is you might have heard about there are some genetic issues where people have brittle bone syndrome. Yeah. And they have that because, what's going on with their genes? If they can't make too much hydroxyapatite, the bone's too bendable. If they can't make enough collagen, for one reason or another, the bone is too brittle. So in people who have brittle bone disease, this is what's wrong. They have a problem making collagen fibers. It's a protein. We make it in the cells. And here they are having a problem. So just moving the wrong way could cause their bones to literally shatter. Ooh, freaky stuff. So that extracellular matrix is really important, whether we're putting down the collagen fibers or we're mineralizing it and making it hard. Okay. We have to have just the right combination of everything for it to work the way it's supposed to. Interesting. You've heard of people with that? You may not know anybody, but you may have heard of brittle bone disease. Yeah. Okay. So if we look into the compact bone, you had a model of this. Okay. Compact bone is made up in a circular pattern. This entire circular structure is called an osteome. It's also called the Habsarian system after Dr. Habsarian, who figured it out, or Haversurs. I can't remember what his name was directly, but it has something to do with his name. It was Havers or Haver, and they named it Haversarium. All right, we're trying to get away from naming things after the people who found them. That used to be the great honor, and we're trying not to do that because it can be confusing. Okay? So we're trying to name them by what they really do. So an osteon is a unit of compact bone. So each one of these round circles is considered an osteon. In the middle of every osteon is going to be a central canal. In that central canal, you're going to have blood vessels. There's also neurons running through the bone too. That's why it hurts when you break it. So they're going to be running through that central canal. The bone cells are laying down the matrix around the central canal in these circular layers that are called lamellae. So they're laying it, the osteoid, the organic part down, the hydroxyapatite, the inorganic material, makes it hard. Now these little osteocytes, because they've now matured, are stuck in these little cavities that are called lacuna. This is when they start to make those cytoplasmic processes, and they are going to stretch out from cell to cell. Right? So there can only be so many of these lamellae because they're far away from the blood supply. So the first cell, right, if here's our central canal with the blood vessels, the first cell is going to pick up materials from the blood, send it to the cells in the second layer, which will then send it to the cells in the third layer and the fourth layer. So there can only be so many of these lamellae around a central canal because we have to keep moving the materials. The further away, the harder it's going to be to get the materials. Once they've gotten the good stuff, the materials they need, the nutrients, and the oxygen, then they have to pass waste products back the other way, from the outermost lamellae to the next, and then the ones that are in the lamellae closest to the central canal are going to dump all the waste products into the blood there. So this is the way these cells can stay alive. Now, there are some blood vessels in the periosteum that's surrounding all of this too. Okay, so it's not just in the central canal, because when you put circles together, they don't fit real closely, and you've got some spaces in between, right? So when we have spaces in between, we still need to get them some nutrients. Now, we have what's called a closed circulatory system. All the blood stays in a vessel from the time it leaves the heart, circulates to the whole body, and returns to the heart. It never leaves a vessel. So now we have hard bone with a central canal. How are we going to connect all of these blood vessels? We're going to have some perforating canals that go from one central canal to another central canal to another central canal until they all join up together with an artery coming in and a vein going out. So we have these perforating, also called Volkmann's canals, after Dr. Volkmann. And then when it gets to the spongy bone, the blood vessels are just going to wind their way through all the trabeculae. So, central canal has all of the blood vessels for the lamellae, the osteocytes and the mellae around it, but all of the central canals are connected by perforating canals. So the blood is always in a vessel. That's the way it has to be. We have some blood vessels that surround the outside of the bone too. When we get to the spongy bone, it's just going to wind its way all through the trabeculae. Yeah, a nice model in there. Was that on the exam? How'd you guys do on that one? Okay? I'm sure she went over it with you? Not really? Oh, you haven't got the exams back. Oh, you won't get them back. They never give you back your lab exams. Yeah, yeah. Because they have like 3 or 4 different versions and they recycle them, so you never get your Scantons back. Sorry. All right, so there it is. So we call it the Osteon or Havasarian system. It's the basic structural unit of the compact bone. We've got that central canal. Because it goes in a circle, we call them concentric lamellae that go around. They're connected by the canaliculi, and then the diagonal connections between the central canals is by the perforating or Volkmann's canals. So if you were working with an old doctor, he'd probably say Havasarian system. If you were working with a young doctor, he'd probably say Osteon system. So it just depends on when you learned it. So now you know both. Same thing with the perforating and Volkmann's. All right, so there they are again, another one. Sometimes I find other pictures I think are better. I'm not sure if this is the one from your book or not. But we've got, lymphatic vessels in here. We've got all kinds of things in here. Blood vessels, veins, arteries, nerves, you name it, everything travels through that central canal. And again, circles don't come together really nicely. Yeah. So we have what are called some interstitial lamellae, right, that fill in the gaps between the osteons. And then usually with compact bone, you're always going to find some layers around the entire outside of the bone, and those are called circumventional lamellae, right? The circumference of a circle, circumventional, right? All the way around. Remember math? Me either. Okay, so that's our compact bone. And just a mention of those. We got to fill in the spaces in between the round circles. They don't fit together nice and tight. So interstitial, and then we're going to put a nice couple layers around the outside of the bone, and that's going to help to keep everything nice and compact and dense and good, right? That's why bone is so strong. Remodeling. Every time we have to remodel, we have to break down osteons and then rebuild things. So they say that that interstitial lamellae in between them is remains of old osteons when we did some remodeling. And you have to do that to grow a bone. Make it longer, make it wider, whatever you need to do. We always have to remodel a bone. We're always going to have some of that fill-in, right? And that fill-in is the old osteons that we remodel. Spongy bone, the trabeculae, again, we mentioned the bone marrow. There really are no osteons, and if there are, they're few and far between, but there really aren't any central canals. Okay. All of the osteons in, or I should say all the osteocytes and osteoblasts that are in spongy bone are close to bone marrow, and there's lots of blood vessels there. So it's not a big deal for them to be far away because there's blood vessels right there. And again, these are good for different lines of stress, right? So up and down stress, sideways stress, you name it. We put our bones through a lot, right? When you sit on a bone, when you're doing all kinds of things, running, jumping, exercising, whatever you're doing, you're putting some stress on the bones. And so between the compact bone and the spongy bone, we can alleviate any problems with breaking bones, because that's the last thing that we want to do is break a bone. So here you go. If we were to look at one of these trabecules and take a little slice through there, we would see this. We do have some lamellae, and that the cells do have canaliculi, and they do communicate back and forth. Okay. But, yeah, there's blood vessels all the way around. So it's easy for them to get their nutrients and get rid of waste. Okay. So this would be just slicing through a little bit of one of these little trabeculae. So a little bit different structure than we see in the compact bone. But remember, spongy bone is always sandwiched in between compact bone. Questions? Everything okay there? All right. Bone marrow, yeah, red bone marrow. We call it hematopoietic tissue because we make red blood cells there and white blood cells. Technically, we call them blood cells. A lot of times when you say blood cells, you're thinking red blood cells, but we also make white blood cells. And hopefully we'll get maybe to talk about that later on. Okay? Again, flat bones, skull, irregular bones, vertebrae, ribs, sternum, part of the pelvic girdle, right? What was that big flat bone there? The pelvic bone, the os coccyx, is made up of 3 bones. Ilium, ischium, and tuberosity. No. Ilium, ischium, and pubis. I'm thinking, do you know what part you sit on? The ischiotuberosity. Yeah, that's the bone you're sitting on right now. That thing in the back there. All right. That's where we make it. And again, once you've reached adulthood, you really don't need to keep adding blood cells. As you're growing, you have to keep adding the amount of blood you have in the body because you're growing up. Once you've reached adult size, though, the red bone marrow and the flat bones is enough to keep making the red blood cells we need. So the marrow in the medullary cavity usually turns to a fat storage. So we call it yellow marrow. And again, this is why you can live for at least 2-3 months without eating. Not that we want to, but you could if you had to. Okay, so making bones. So this is going to be something new. Okay. In the human fetus and infant, bones develop by 2 methods. Right? Intramembranous ossification or endochondral ossification. Most of our flat bones are made through intramembranous ossification. All of our long bones are made by endochondral classification. When we talk about cartilage, the cells that make up cartilage are called chondrocytes. Chondrocytes. Do we remember that? When we talked about tissues? So endochondral, chondrocytes, cartilage model, membranous, from a membrane. Okay. So kind of breaking down those words, makes it a little bit easier. So endochondral ossification means we had a cartilage model. Intramembranous ossification means it happened from a membrane. Okay. Intramembranous. We do have a little bit of a template, we could say. It's not really a model, but it does give us a little bit of some structure to kind of start making spongy bone. Yeah. So it's connective tissue. Right? There are some special cells in there. and these are during development, okay, so the cells change over time. These are like some stem cells that have been given a signal, and they become mesenchymal cells. There's a word for you. Mesenchymal cells. Okay? And they start to produce that fibrous connective tissue that's going to kind of give us a little template as to where we're going to make spongy bone. They start to cluster together in to what we call an ossification center, where they become osteoblasts. So the mesenchymal cells are a type of stem cell. And differentiate means it changes from a stem cell into an osteoblast. You know, they talk about embryonic stem cells and adult stem cells. The difference between them is that embryonic stem cells are not differentiated. They haven't been told what kind of stem cell they're going to become. Once you've reached a certain period of time in your life, and they call it adult stem cells. I'm not so sure it's adult, but it's sometime between, probably 5 to 20 years, 18 years old. All of these stem cells have been told this is the kind of stem cell you're going to be. You're going to be a stem cell in the stratum basale, which means you're only going to make skin cells. You're going to become an osteogenic stem cell. You're only going to make bone cells. You're going to become a hematopoietic stem cell. You're only going to make red blood cells. Okay, so at some point during our development, those embryonic stem cells that could become any kind of stem cell has been told this is the kind of stem cell you're going to be. And that's what we mean when we say differentiated. It can no longer become a bone cell if it's a blood cell. It can no longer become a skin cell if it's a bone cell. Okay? It's already been programmed to be a specific kind of stem cell. And it can't revert back to do anything else. If they could, then we would be growing new organs whenever we had problems, but they can't. Okay? They're trying to get them to do that, but maybe someday they will. They'll give you an injection of something, and if you're having a kidney problem, you'll just grow a new kidney. How cool would that be? But we're not there. Right? So these mesenchymal cells are a type of stem cell that are going to migrate, create the fibrous connective tissue, and then some of them are going to differentiate into osteoblasts and start making the organic matrix, the osteoid, the collagen fibers. They're going to be trapped, we already know that, and then blood vessels are going to start to grow into those areas. The hydroxyapatite is going to be laid down, and now they're trapped and they're osteocytes. And that's basically how we make flat bones. So, some pictures, right? So, we've got some mesenchyme cells. They're going to start to kind of collect, cluster, and they're going to turn into osteoblasts. They start to lay down the fibers, the blood vessels are there. They're laying down the collagen fibers and all the organic parts of the matrix, and then it becomes hardened. They completely become entrapped, okay. And we get that kind of honeycombed or trabeculae look, spongy look. In between the spaces, we're going to start filling it in with bone marrow, and we will develop some compact bone on the outside of it, eventually. Okay. So calcification, mineralization, hydroxyapatite are how we are going to make the matrix hard. Same thing happens both in spongy bone and compact bone. And we keep doing it and doing it and doing it until we've made the bone. Until the bone is at the appropriate size. So there you go. They're making canaliculi. They're putting down the calcification, right? The hydroxyapatite and all the other minerals come in. So now we're making a little beam. We're making a little trabeculae. They are trapped. They put out the processes. They mature. Same thing. And we keep making it and making it and making it within the membranous tissue. There you go. Eventually, it's all made. We got lots of blood vessels. We're going to fill it up with red bone marrow. Right? And you remember, this is happening during development. Okay? So everything's not in place yet. We're making it. It starts in an embryo, and then it's going to continue into childhood, right? As the bones grow. Some bones are going to reach their adult size sooner than others, right? You ever notice like 2-year-olds and 3-year-olds somewhere in that age where their heads look huge and their bodies look tiny? Yeah. So these skull bones are going to be some of the first ones to reach adult size. It's true. That's why little kids have more of a problem with drowning, because their heads are so heavy. You have to be careful, because they can't lift themselves up. Okay. There's little bits of information you pick up along the way. Right. So go ahead, go out and look at the 2 and 3-year-olds and look how big their heads are. Whoa, look at the head on that kid. My nephew was that way. I mean, he eventually fit into it, but his head was huge. All right, there you go. Now, what about the long bones? Well, we have to make them by a different process. Endochondral ossification, which means we need a cartilage model. The model has to tell us how to start making that bone. So your femur, your ulna, your radius, your tibia, your fibula, your humerus, your phalanges. These are all going to be made with a little bit of a model. Okay? So, again, mesenchymal cells. Okay? These are the cells that are going to develop into chondroblasts this time. Cartilage cells. And the cartilage cells start to put down cartilage as a model. They secrete an extracellular matrix. It's not calcified yet. It's, a little more gel-like. And again, the chondrocytes are going to become trapped in a little lacunae. Only there's no canaliculi connecting them. So they have to rely on a perichondrium, an outer layer, that's going to supply them with their nutrients and take away waste, because the perichondrium will have some blood vessels in it. There you go. This is a cartilage model. It's got a perichondrium around it, which has blood vessels in it, which is supplying the cartilage cells with everything they need, because they're trapped too. At some point in development, we're going to get those cartilage cells calcified. Once they're calcified, we're going to create, because blood vessels will be able to enter into this area now, a primary ossification center. It starts with making some spongy bone covered by compact bone. So, the mesenchymal cells, instead of becoming osteoblasts, like they do in intramembranous ossification, become chondroblasts, cartilage cells. And it starts to make this model. It's not complete, obviously. The model will continue to expand and change as we are making the various bones. That primary ossification center is going to form in the center where the chondroblast, the cartilage, has become ossified. Yeah. Woo! Become hard. And then we can bring in some blood vessels into that area. Along with the blood vessels are going to come the osteogenic cells, which become the osteoblasts. And they start to put down bone. The chondrocytes are going to die as they become ossified. The perichondrium, once again, we had stem cells in there, is going to become a periosteum with the osteogenic cells that become osteoblasts. Back to the picture. Okay. So we're here, right? The cartilage cells become ossified. They start to die off. Now we can start to penetrate into this area with arteries and nutrients, right? And veins and everything else we need. As they become ossified, the chondroblasts die. Chondrocytes, chondroblasts, they're dying in this area. They're still alive up here though. So they're still making more cartilage towards the ends. But in the center, we're starting to now put down bone. The perichondrium in this area becomes the periosteum. The periosteum, osteogenic cells that become osteoblasts, that become osteocytes as we start to ossify them or calcify them, mineralize them, make them hard, put down the hydroxyapatite. So more chondrocytes become ossified, more die, the bone starts to spread towards the ends. So we're kind of building those long bones from the center outward. This is the primary ossification center, starting in a cartilage model. Keeps replacing the chondrocytes that have become ossified by this other coloration here, and they die. But the ones above it are alive, and they keep growing the bone longer and longer and longer and longer, because it keeps making more and more and more and more cartilage. And it's kind of like the bone is chasing the cartilage out towards the ends. And that's what's making the shaft, the diaphyses, longer and longer and longer. Okay. At some point, a secondary ossification center appears in the two epiphyses. This is only going to show you one epiphysis, but it happens in both, at both ends of the bone. Okay. From the center, the shaft, the diaphyses, growth is continuing this way, and now we're going to start to grow the epiphyses, the head of the humerus, the head of the femur, the epicondyles down at the bottom. In between these two ossification centers, we have live cartilage that keeps making cartilage model for the bone to follow. So here you go. We're making the bone. We're making more cartilage. We get a secondary ossification center in the epiphyses. We still have live cartilage, right? But this is becoming more and more calcified. The shaft is getting longer. The epiphyses is getting bigger. And eventually, we get to this plate here where all we have is what's remaining of the cartilage. Once all that cartilage is used up, dead, the epiphyses and the diaphyses join. But we could see this, it's almost like a little scar where the epiphyseal plate was, as these two portions of the bone get joined together. So we start in the middle, we're growing that shaft, that diaphyses longer and longer and longer. At some point, we start a secondary ossification center in the epiphyses on both ends. That's just showing you one end, right? Again, we had a model, and now we're going to start to follow the cartilage model by putting down bone and bone and bone. Eventually, what's left on the top here will become the articular cartilage on the ends of the epiphyses. And the cartilage here becomes sort of like a little line that we can see where the epiphyses and the diaphyses have connected, and we call that the epiphyseal plate. And then this will keep continuing until bone growth has completed. And that's usually by the age of 25, your long bones have completed growing. It might be sooner in some people, but 25 is the upper range that we've been told is where long bones are done growing. They will not grow anymore. So here's a picture kind of taking us through the whole thing. Okay? And there's the epiphyseal plate while there's still cartilage. Once they've joined together, we just call it a line because there's no longer any cartilage there. And our bone is complete. Pretty cool. So we had a model from the time we were tiny little babies. Yeah, so again, by the end of a long bone growth, that cartilage is limited to the articular cartilage on both ends, and then we do have the epiphyseal plate, which becomes the epiphyseal line. As long as we have the plate, growth is possible. But once all that's gone, yeah, we kind of have the epiphyseal plate, all the cartilage is dying, the epiphyses and the diaphyses, that little gap between them kind of closes and they're now connected to each other. And then the primary and secondary marrow cavities are all united into one single cavity, and the bone can't grow anymore. And we have a long bone. Look at that. Woo! 12 weeks. All the red is cartilage. Yeah. Because we don't want that to be bone yet, because it's got to still come out. Right? Yeah. Those of you who have children know exactly what I'm talking about. Those of you who haven't, just wait and see. And you guys, your wife will let you know. All right. Remodeling. Yeah. Bone remodeling occurs throughout your life. Yeah. We reabsorb, we deposit new bone. Yeah. Again, 10 to 20% of your skeleton gets replaced every year. Oh my gosh. I bet you've had more micro fractures than you can shake a stick at. How many times have you hit your shin? Got a nice bruise. Oh, microfracture. Banged your hands, your fingers, right? Yeah, little tiny little fracture. You don't need to go to the doctor. You don't need a cast or anything, but you've broken the bone a little bit. Yeah. And then, any weightlifters? Nobody's owning up. Okay. There's something called Wolff's Law of Bone. He said that bones reshape through use and disuse. Mechanical stress. Okay? If you're a weightlifter and you're building those huge muscles, that'd snap your bones in half if your bone didn't get wider. It doesn't get longer, but it gets wider to withstand the strength of that muscle. It's true. Use it or lose it. Anybody who's ever had a broken bone and it's in a cast, right? They haven't been using the muscles or the bone because it's been repairing. Yeah, the muscles kind of atrophy a little bit. Right? Yeah. And then you start using it again. Your bone has to get a little wider to withstand the stress those muscles are putting on you. You know, this is one of those places where they say form follows function. How many people slouch? Yeah. You're going to look like this when you're old. Your bones are remodeling. Stand up nice and straight. We all slouch from time to time. But if you're a constant sloucher, by the time you're my age, you're going to be walking like this. There are other reasons, things that happen to the bones, but yeah. I mean, your muscles are pulling on your bones in a certain way. That's what's going to happen. How many people are right-handed? Do you know that your right bone is bigger than your left bones? If you're left-handed, it's going to be opposite. People who, like the football kickers who kick with their right legs, stronger bones, stronger muscles. Unless they're working out evenly. And that's when you should work your left side of your body as well as your right side of your body. Because form follows function. So this is just a funky little picture I took from the internet. Okay, there's this website if you're interested. It just shows. All right, so let's just say, your bone's fine. Now you hit your shin, and you've got a little microfracture, right? It's not anything major. It hurts. You probably bruised it and that sort of thing. But you probably got a little microfracture there. And so osteoclasts come in. They look like, Pac-Man? And they clean up all the cellular debris, and they clean up and they dissolve any jagged little edges or anything so that we can put down some new bone. So osteoblasts will come in, okay, and they start to replace the bone. But again, if we look on an x-ray, we can see where that remodeling has occurred, right? Where the fracture's been filled in. So, use and disuse. So, microfractures. And this happens too if you have a major fracture, but with a major fracture, you need to have it set and you need to have casts and stuff like that. Okay. One of the important things that happens with bones is calcium homeostasis. Calcium and phosphate are used for a lot more than just bone structure, right? Phosphate is part of DNA, RNA, ATP, phospholipids, and those buffers that we have in the body to help prevent pH changes. So we need phosphate for a lot of things. And the hydroxyapatite is calcium phosphate. So we're storing a lot of that in the bones. Calcium is needed for your neurons to communicate with each other, for muscle contractions, for blood clotting, and for exocytosis, to get things out of the cell. So again, we think of calcium in bones, but yeah, in phosphate and ATP and in the cell membranes with the phospholipids, but we need it for a lot of stuff. And so we can actually deposit extra minerals in the skeleton and then withdraw them if we need them for other purposes. If for some reason your calcium level in the blood gets too low, you'll start to release some of that calcium via the osteoclasts from the bones to increase your blood calcium level, because you need calcium to make your neurons work. Without it, they don't work. Okay, so we regulate it. About 98% of the body calcium reserve is in your bones. Okay? And again, homeostasis, maintaining the consistency of the blood calcium levels. We've mentioned why we need it, and we can literally, excuse me, kind of mobilize calcium. Put it into the bones, take it out of the bones. Put it into the bones, take it out of the bones. Take it into the bones, take it out of the bones. Okay? So we're constantly monitoring the calcium level and making sure it's where it should be, and we're constantly moving it in and out. There are 3 hormones that we use to regulate blood calcium level. Okay? Again, intake, output, so what you take in your food, what you get rid of in the urinary tract and the digestive tract. Normally what goes in helps to balance what goes out. But if we're not taking in enough calcium, then we need to go to our osseous tissue. We need to go to bones. We need to start breaking the bone down, dissolving the bone to keep the calcium level where we need it to be. Right? That's why you usually drink calcium modified milk. Milk with vitamin D and milk with calcium, because this is going to help us to maintain calcium levels. So we have calcitonin, calcitonin, and parathyroid hormone. These are 3 hormones that are going to regulate calcium homeostasis. Calcitonin is vitamin D. We need the skin, we need the liver, and we need the kidneys to make calcitonin, the most potent form of vitamin D. Starts in our skin. What does it do? It's a hormone that helps us to raise calcium levels. It causes your intestines to take up more calcium from your foods. It increases calcium reabsorption, breaking down the bone in the skeleton. It works on the kidney a little bit, but not a lot. We can reabsorb certain things from urine that we need, and so it can promote some of that reabsorption from the urine by the kidney, but it's not super strong. We also need it if we want to make bone. We have to have adequate calcium and phosphate in order to make bone. Inadequate amounts of calcitriol usually results in the abnormal softness in bones. Rickets. Rickets. Sailors in the old days, on the old sailing vessels, long before we had modern stuff, would be out at sea for a long time. They didn't have refrigeration. So the foods they could take were either gotten by wherever they were going or dried foods, right? They didn't have a lot of citrus fruit. So they would develop rickets, soft bones. And a lot of times rickets is shown by that kind of exaggerated walk on the long bones of your legs. Okay. So at some point, somebody figured this out, a doctor, and they sent them with limes, because limes seem to last longer. And so that's why they called the British Navy the limeys. You never heard that? Ever heard that? All right. Well, yeah. In adults, we call it osteomalacia. This normally, we get enough calcium in our diets nowadays that we don't really have this problem. But in poorer countries where they don't have access to these things, this still occurs. Okay. All right, so keratinocytes in the skin convert cholesterol derivative into vitamin D precursor in the presence of UV radiation. So we need to have a little bit of sun. That precursor then goes to the liver to become another intermediate form or precursor, which finally makes its way to the kidney and becomes calcitrol only when we release parathyroid hormone. So it's a complicated process. You don't need to know the whole process, okay? So don't forget. But it's a 3-step process in order for us to do this. Most people in Michigan have a vitamin D deficiency because of the winter months. Number 1, you're bundled up because it's cold. Number 2, it's usually gray and the sun doesn't shine. And you don't spend a lot of time outside because it's cold. You should be taking a vitamin D supplement so that you can keep your bones in good order. So this is just a little picture. Okay? We start with a cholesterol derivative. In the skin, it gets turned into this precursor. In the liver, it gets turned into another precursor. And then finally, in the kidneys, in the presence of parathyroid hormone, it becomes calcitrol. Helps with bone reabsorption, taking the bone out, reduces excretion of calcium or reabsorb calcium in the kidneys. Also, it helps our intestinal tract and our stomach to absorb more calcium out of our diet. So one way we get calcium. The other way, well, don't worry about this one, okay? The thyroid gland. On the back side of the thyroid gland are 4 little spots that are called parathyroids. Oh wait, I'm on the wrong one. I'm on the thyroid gland. All right, let me go to that one first because I talked about it. Okay, the parathyroid gland. This is on the back side of the thyroid gland. There are 4 little areas, right? And it releases one hormone that's called parathyroid hormone. And this is going to be the primary regulator of calcium homeostasis. It stimulates the osteoclasts. What are they going to do? Break down the bone matrix to increase blood calcium levels. So what tells us osteoclasts to go into action when calcium levels are low? Here you go. We're going to release parathyroid hormone. It also increases renal absorption, taking back more calcium, right, from the kidneys, in the kidneys from the urine, and it can also help to stimulate more vitamin D synthesis, starting in the skin. So here we are. This is the thyroid gland. This is the back side, the posterior side. There are 4 areas like this. 1, 2, 3. I don't see the one over here. At least I can't find it on this picture. And that's where we're going to release the parathyroid hormone. The thyroid gland produces calcitonin. Okay. What is calcitonin going to do? Did I not put it in there? I didn't. Here it is, calcitonin. I said skip it. Never mind. I'm losing my mind. I'm trying to go too fast. So calcitonin is produced by the thyroid gland, and what it does is when blood calcium levels are too high, right, it's going to stimulate deposition by the osteoblasts, and it inhibits the osteoclasts. Losing my brain today. I apologize. So, Calcitrol, skin, liver, kidneys. Maintain blood calcium level when it's too low. Okay. Calcitonin, thyroid gland, when levels are too high. Deposit the extra calcium in the bone. And that's your thyroid gland on the front, the anterior side of it. Okay. And they're showing it here. Parathyroid, on the back of the thyroid gland, 4 areas that release parathyroid hormone, stimulating osteoclasts to break down the bone matrix when calcium levels are too low. So you've got 2 for low, 1 for high. Calcitrol and parathyroid hormone, too low. Calcitonin, too high. And again, homeostasis, keeping the balance right where it needs to be. You don't need to know how we make the calcitrol. Okay? Just, I wanted you to see how we get there. Why do you need sunlight on your skin? This is why. All right, we do have some names when we have calcium imbalances. Hypocalcemia is a deficiency, right? It changes membrane potentials and causes overly excitable nervous system, and tetany, which is muscle spasm, if you don't have enough. Okay? You can also get laryngospasm, which is here in your throat, those muscles around the throat area, which can cause suffocation because you can't breathe. Closes off your larynx. The causes vitamin D deficiency, diarrhea, thyroid tumors, underactive parathyroid glands. These are all conditions that are usually monitored by doctors. Pregnancy and lactation can also increase this because you're giving a lot of that calcium to the baby. Why? The baby still has endochondral bone formation occurring. Bones aren't grown yet, taking all the calcium from mom. So, again, doctors regulate this, and they'll make women who've just given birth take extra calcium and things like that. You take it when you get older, too, because you have a tendency not to be as efficient when you're older. Hypercalcemia, excessive calcium levels make ion channels less responsive, and your nerves and your muscles are less excitable, meaning you can't move them as well, and you can't think as well either. It can cause emotional disturbances, muscle weakness, sluggish reflexes, cardiac arrest. Your heart stops pumping. It's a muscle. It's very rare that it occurs, but it is something that has been seen. All right. Bone fractures? Okay, I'm going to let you look at this on your own. Okay, we have different types that we can have. Ouch, yes. Bending, you name it, there's all kinds of things that we can do to our bones. And so we have a couple phases. It takes anywhere from 8 to 12 weeks for a break to occur. But in the elderly, like me, yeah, you hear about hip fractures and things like that. It takes a long time for these fractures in the elderly to heal, okay? But, you know, you get a hematoma because there's blood vessels in the blood, so you're going to get bleeding. But then you get a fibrocartilage callus that forms around the break, and then you're going to get a bony callus, okay? And after that, we go through bone remodeling. We've got to do osteoclasts to get rid of all the jagged pieces and broken pieces, and then osteoblasts come in and put down the new bone, okay? So here's a little picture from your book that goes through it. When you've broken the bone, it's going to bleed. Okay? There's blood vessels in the bone. you're going to get a soft callus, they call it. It's fibrocartilage that comes in and starts to kind of hold the bones together. Obviously, we're going to get new blood vessels to start working. The next thing that happens is you get a hard callus that replaces the fibrocartilage on the outside. Spongy bone, this is a long bone, so spongy bone starts to grow on the inside first. And then finally, all the little small bone fragments and everything are going to be removed by the osteoclast. They'll be working over here too. And then we're eventually going to replace the bone. But you can see where bone's been broken because of this, right? That hard callus that forms. So when a bone has been broken and it heals, you can see where the bone has healed. Okay. So you can read through that if you have more questions about it. I'm not going to test you on it. I just thought it'd be interesting to talk about. But again, you know, how do you treat fractures? Well, they have a closed reduction. The bone fragments are kind of manipulated back into their normal position without surgery, and you put a cast on it. And the person has to stay that way for at least 6 weeks, maybe longer, depending on how bad the break was. An open reduction, unfortunately, this is usually when it's broken the skin, and they have to go in, and a lot of times they have to put in some plates, some screws, or pins to realign the fragment because it's really pretty severe. And again, normally a cast is going to be used in this case also. All right. And so when they take the cast off at 6 weeks, it may not be completely healed because it takes anywhere from 8 to 12 weeks for full healing. So, and the more severe the break, the longer it's going to take to fix. So here's one where they had to put in a plate and some screws to hold everything together because it was a really bad break. Okay. You've had that?
I have
You have? Now? Oh, okay. Yeah? Ouch. What'd you do to break it? Can we ask or no?
I got into a car accident.
Car accident?
I didn't know it was broken until I tried to get out when I got out.
Oh, gosh. Yeah. Just a matter of interest. You know how some people ride in the car with their feet up on the dashboard? These two girls were in a car accident, and one of them did that. The dislocation of the femurs was unfreaking believable. Look on the internet for feet, car accident with feet on dashboard. I mean, this pelvic bone was all the way over here. It was unbelievable. Scary stuff. Yes. All right, just as you get older, you get osteoporosis. Yeah. It's the most common bone disease, you know, as we get older, we start to lose bone mass as early as 35. And by the time we reach 70, we've probably lost about 30% of our bone mass. So again, staying active, using your muscles, doing a little workout, lifting some weights, not tons of weights, but you know, but staying active. Osteoporosis usually affects the spongy bone a lot more, and after a while we start to get fractures of the hip, the wrist, the vertebral column, all kinds of things can happen. That spongy bone gets brittle like this, okay, and that's not good. So good diet, making sure you have calcium and vitamin D to keep your bones nice and strong throughout your life. But activity is also going to help. Okay? And there are just some things with aging that you can't get rid of. Here's another picture. Here's somebody who's broken their vertebrae. Ooh, scary stuff. Okay, but osteoporosis, your spongy bone is breaking down. Okay? All right. On Thursday, we're doing muscle. I'm not going to go through all the kinds of muscles. We're just going to get into how a muscle works. Okay? So I'm thinking next Thursday, we have our second quiz. Next week, Thursday. Okay? So think about that. Look at your schedules. And let me know on this Thursday if that's going to work. If not, we can put it off until the following Tuesday. That's cool too, and we'll start moving forward. I want to get you on the new material as quickly as possible, okay? And I did speak with Linnea, and she's going to give you some heads up too in lab, okay? So you'll probably hear it there first, and then we'll go over it again in here, okay? I'm sorry, we're being behind, it's my fault. I need to cut my PowerPoints down. But it's also interesting. I'm sorry. It's me.
So what will the quiz next week be on?
6, skin, bone, and muscle. 11. I think it's 6, 7, and 11. Yeah. Okay. But let me know if that works with everybody's schedule. And I'm willing to work with you. If it doesn't, you can take it over in the testing center. We'll figure something out. But I'd like us to get back on track. Okay? So that we will. What? I don't know. Get back to normal. I don't want us to get too far off of what we did in the lab. And yes, you can blame me. I did it. It's all my fault. I'm sorry?
Is it too late to look at that either one?
No.
I mean, it's like
Arnella, can you hang out for a second? Oh, I took it out already. I'm sorry. You're welcome.
So do you want to take a look at it or no? You can have them like until like 2 o'clock. You can do it today or you can do it Thursday. Thursday. You want to do it Thursday? Okay.
Yeah, thank you.
All right. Did anybody not get their test back? Who's here? Who are you? There you go. Thank you. Okay. Sorry about that. I think I skipped that one when I was calling names before. I apologize. Yell at me, and I will search for things. Take it for
So, their tricky is something on there or similar areas. What else did you learn?
I didn't like that you couldn't touch the bones. Cause I mean
Easier when you can point to what names on and just
yeah, cause
Okay,
if I'm
well, okay,
standing up,
so besides the rules, you can complain about the rules all you want, but it's not going to change the rules because I don't make the rules, nor do I set up the lab exam. This is not my baby, okay?
I don't know.
Don't shoot the messenger. You can complain to the messenger all you want, but it's not going to change anything. Sorry. But that said,
I would learn to do actually
yeah.
study these damn things more.
There you go. Yeah, study those damn things more. Study those posters. So this is what I want. What did you learn? How are you going to take what you learned forward so that you can have more success next time? So don't forget about the posters. Take pictures of stuff. So now that you know how they arrow everything, right? What else did you learn?
Study each section day by day.
Study a little bit every day.
Going to the library and using all the stuff there that's in the room is very helpful.
Yeah, very helpful. Go to the library.
I got another question.
Sure.
When they do the practice exam in the library, why they don't do it like resemble the same thing in here? Why is it on paper?
I don't know. I don't know. Maybe because they want to leave all of the anatomy, models for people to study. They don't want to tie one up or tie a couple up for a practice lab exam.
Okay.
So that's my guess. So, all right, so going forward, now we're doing nervous system. I do have an extra credit option on for your next lab exam. Okay? You need extra credit.
Yeah, okay.
That's very helpful.
What is the extra credit? I'll have to look. I don't remember how much extra credit it is, but it's associated with the brain. Okay? You get to make a brain hat.
What?
You make a brain hat. And if you bring your completed brain hat to the lab on our next lab exam, so I should have a whole bunch of brain hats up here, so I will give you extra credit for it. But you have to have all the different parts of that brain hat complete. Okay? This will help in your studying of the brain.
Do you have an example? Yeah. Is it like a brain model that we're making?
Just chill. Goodness gracious.
Do you have this for Oh, is this the
You can't. We got scatterbrain.
Now, if I get a pile of brain parts, it's scatterbrain. So this is completely optional, hence it's the extra credit nature. It is 3 pages on cardstock. If you look, you see the left hemisphere, and then you see the right hemisphere. If you choose to make a brain hat, you cut out the brain, okay? And then you cut on the dotted lines, and you glue or tape your brain together. As you do this, it will form literally a hat. It'll be curved, and then you tape both pieces together. You make a brain hat, okay? But the instructions are on the first page. You need to label the following structures on the brain. So you're going to label everything. You're going to color each lobe a different color. And I think that's it. It's coloring and labeling. Okay? I don't remember. I have to look. No, I think it was like 5 last semester.
How much of that helps for color?
Quite a bit, like half a grade.
Oh, okay.
So, I mean, if you had a 70, if you had a C minus, 5 points extra credit, that's a 75. You go, that's like a partial letter grade. But, so you have to have it physically here in order to get 5 points. I heard everything. Left it in my car. Okay, how fast can you run? Right? I left it in my car. Can I still get extra credit? How fast can you run? I left it on my kitchen table. Sorry. It's at home, but here's a picture of it. I made it. Look, here's a picture. So here's a picture of me. I'm wearing it. Okay, that's great, but it's not physically here. I'll give you some points, like maybe 2 for completing it, or 2.5. I'll give you half credit. But in order to get full 5 points, it's got to be right here. Okay? If it's on my kitchen. Kid you not, this happened. It's on my kitchen table. Okay, show me a picture. Oh, I took a picture. No, forgot I don't have my picture. Do you think it was really done? No extra credit. Okay. At least take a picture of it on your phone. But for full credit, it has to be right here. Sound good? This is your extra credit. It will be very helpful when you're learning all the different parts of the brain. Okay? So, we're not going to get into, I'm not going to give you the brain stuff, I think, until next week. Maybe I'll give you my brain sheets on Thursday if I get them printed in time. But obviously you have your lab books, and that's the end all be all. So if you want to start on the brain today, you can do that. So, just my lab sheets that I print out. I don't know if you're ready. All right, so questions before I go on?
When's this due?
Lab exam. Second lab
Oh,
exam.
sorry.
Good deal? Cool. All right, so now, nervous system. Now, I am challenged in teaching this lab because you guys haven't done the nervous system yet. There we go. But that's all right. We'll make it work. So you guys get the first 20 minutes of my lecture. So as you're going through this business, at least of the nervous tissue, you know what the heck you're looking at, okay? So, in general, the nervous system has three major functions. It serves a sensory function. So in this lab, you're going to see like sensory type nerves. It has an integrative function. That means it's thinking about or processing that information. And it has a motor function. So there's some sort of output of that nervous system. Okay? Hold on a minute, guys, in the back. I have to flip my slide. Now, there's two major subcategories of the nervous system. There's the central nervous system. That is the brain and the spinal cord. And then the peripheral nervous system, those are your nerves coming out from that brain and spinal cord area. Okay? So you've got cranial nerves, spinal nerves, ganglia, all that business is in your peripheral nervous system. So you'll need to know the difference in lab. What is what? Okay? Some terminology that you'll see. And I don't know if it's on this one. I know I put this terminology on the spinal cord one that's at the print shop right now. Is the difference between, or two different words, sensory and afferent. Okay? So your peripheral nerves are going to sense things from the environment. So I'm going to touch this and I'm going to sense that it's smooth. I'm also going to sense that it's cold. Maybe you're sensing that if there's a feather tickling your face. That's all sensory information. It goes through sensory nerves. These nerves are also called afferent. Okay? That's very important. Your afferent nerves carry sensory information. Okay? Think about it this way. When you hear something, you're hearing something from the environment. Okay? That's a type of sense. Imagine you have your kid is being very obnoxious, or you have a very annoying, like, younger sibling. And what do? Hey, 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 trying to get your attention, right? Hey, 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 hey. You're sensing that. The afferent division is sensing that obnoxious sibling. Don't need that right now. Okay. The types of nerves that make you respond to the environment are your motor neurons. Okay? Again, you're going to see more of this on Thursday when I give you your spinal cord information, reflex information. But your motor nerves are also called your efferent nerves. E-F-F-E-R-E-N-T. Sorry, it's not on this board. This slide just reminded me of it. So, efferent nerves carry motor information out. Okay? So that annoying sibling that's going, hey, 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 your motor response to that annoying sibling is saying, enough! Okay? Efferent is your motor response. Okay? Now there are also interneurons. These interneurons here are found in the central nervous system, in your brain, and in your spinal cord. Generally speaking, they're pretty small neurons, and they're used for processing. They're the intermediary between the sensory and motor nerves. Okay? So you have sensory information in, transmitting that signal to an interneuron, and then that interneuron is going to send that signal to some sort of motor neuron output. Okay? Afferent in, interneuron in the middle, efferent out. general neuron structure. Okay. Some things to note. Oh, that's gonna be a mess. All right. Entertain yourself, if you will, and draw yourself a neuron. We're gonna draw Medusa on a bad hair day here. And a long portion. And some legs down here. There you go. Draw yourself a neuron. So, now, these neurons look like your interneurons. Some neurons look quite substantially different than this, but they all have similar parts. Okay? This is your cell body. It's also called your soma. This one has a third name, the parakaryon. P-E-R-E-I. 3 names, same structure. Parakaryon. Karyon. Maybe I didn't spell it right. P-E-R-I-K-A-R-Y-O-N. Okay, good. So this whole thing, should I say, is the body. These are dendrites. The pointy outy things of the cell body. What's that? Yeah, kind of like the fingers. Medusa's bad hair day. Okay. Dendrites receive information. They're big for receiving information either from the environment or from another neuron. Okay. So you've got these dendrites coming out. You've got the soma. What else am I forgetting? Inside this body, what I should have done is done this. Soma. You'll find a nucleus that contains the DNA. Okay. You'll also see rough endoplasmic reticulum. You guys know what rough endoplasmic reticulum is? Please tell me yes. This rough endoplasmic reticulum in neurons are called Nissl bodies. N-I-S-L-B-O-D-I-E-S. So these Nissl bodies are making all manner of substances that are being used by the neuron. It can even make neurotransmitters that the neuron is going to use to chemically communicate with another cell. Okay, then we have these neurofibrils. Neurofibrils. Oh, wait, let me back up just a little bit. This portion here is your axon. That big portion there is the axon of your nerves. Nerve of your neuron. Nerve transmission, so action potentials, signals are going to move in one direction down this axon. Okay. What also moves down this axon are neurotransmitters. Neurotransmitters are chemicals, again, that these neurons use to communicate with each other. They are made up here, but they're used way down there. So neurofibrils, N-E-U-R-O, are what make up the axon of a neuron. They're like tracks of, they're like cart tracks in the caves. You know, you go in caving or mining, and you can get in the little buckets and go from one area of the cave or the mine to the other area of the cave or the mine. That's kind of what this is like. They're tracks that will send information from the soma down to the terminals. Or not information, it'll send substances like neurotransmitter from the soma to the terminals. Okay? So the axon is made up of neurofibrils. Now, in some neurons, but not all, you might see them wrapped. You might see wrapping around them, like this. This wrapping is a myelin sheath. It helps to insulate that axon. It allows for faster transmission of nerve impulses. In the peripheral nervous system is made of, this myelin sheath is made of Schwann cells. And I'll show you a picture in a little bit of these Schwann cells. It is literally a different type of cell that wraps itself around hundreds of times around the axon. Okay? And a lot of these Schwann cells will wrap themselves around these axons. There are small areas of the axon with no myelin sheath. This is called the node of Ranvier. When we have very fast, we just did this in my lecture today, when we have very fast communication of nerve impulses, that communication transmission is going to happen at one of those nodes of Ranvier. All right, and then down here, we have your terminal region of the neuron, your synaptic terminals. I guess let's call them, your book calls them axon terminals. So that nerve impulse is going to go from here, down the axon, down here, and be transmitted to another cell down there at that axon terminal. Okay? At the very end, that portion is what will interface with another cell. That's your synaptic knob or synaptic terminal. We have a blow up of a synaptic terminal here. Okay? So these, for example, are all your neurotransmitter vesicles. So synaptic knob, there's a lot of mitochondria, neurofibrils in here. You can take a look at this and pair it with your lab book. I think that's the gist of a neuron. dendrites, the soma, nasal bodies. This is your axon. Bless you, I think. This is the myelination. These are your Schwann cells that form that myelination. And here's your axon terminals with the synaptic end bulb. You might see the term glia or glial cells. So there are neurons, and then there are glial or helper cells. Glia is like, almost like glue in some instances, at least in your central nervous system. They help hold the neurons together. They help protect the neurons. You don't need to know these for lab anyway. I talk about them in lecture. There are two types of neuroglial cells in the peripheral nervous system. The one that you're going to want to know are these Schwann cells, because they're the ones that form that myelination sheath around the axons. This is the Schwann cell. Here's the nucleus of the Schwann cell. It literally wraps itself around the axon. Here it is. This is the formation of that myelin sheath. Here's your Schwann cell. Here's the axon. It engulfs the axon and starts wrapping around it. The wrapping gets more and more and more and more, and pretty soon you have this really thick myelin sheath that surrounds it. So this myelin sheath is mostly phospholipids, because it's membrane, and protein, because a cell's membrane is made of phospholipids and protein. So those help speed up transmission of a nerve signal. Physiology. That's what we did today. What else do you guys need to know before I let you go? Self-story connection. Oh! This is another example of your synaptic knob endings. So this is the terminals of one axon. This perhaps is the dendrite or soma of another neuron. We have a nerve impulse coming down. That's going to cause these vesicles with the neurotransmitter to release the neurotransmitter into the synaptic cleft. A neurotransmitter is just a type of chemical. That chemical is going to go through the cleft. It's going to join with some of these receptor proteins. And once joined, the receptor proteins are going to open, and we have other ions flowing in that hopefully then are going to create another action potential to continue that electrical signal forward. Continue it along another neuron, or continue it into like a muscle fiber or something along those lines to affect some sort of change. Okay? Let me look at your list here. Somebody know fiber tract. The only thing I did not mention is this idea of a tract. And a tract is a group of neurons. Okay? So you'll see tracts more when you get into the brain. Ascending tracts and descending tracts are groups of neurons. Also note that the difference between a neuron and a nerve, they aren't interchangeable. A neuron is one nerve cell. A nerve is a group of neurons. Okay? Questions? That is nervous tissue in 20 minutes. Crazy flyby. So hopefully Professor Sack will get a little more detailed than I just did, but I just don't have the time. So now I'm going to let you go. I at least gave you stuff to get started with nervous tissue.
Good luck with today's lesson.